Figure 7.3, Variations in Meiosis. By the end of this section, you will be able to explain how non-disjunction leads to differences in chromosome number and describe how errors in chromosome structure occur through inversions and translocations. Inherited disorders can arise when chromosomes behave abnormally during meiosis. Chromosome disorders can be divided into two categories: abnormalities in chromosome number and chromosome structural arrangements. Because even small segments of chromosomes can span many genes, chromosomal disorders are characteristically dramatic and often fatal. Disorders in chromosome number. The isolation and microscopic observation of chromosomes forms the basis of cytogenetics and is the primary method by which clinicians detect chromosomal abnormalities in humans. A karyotype is the number and appearance of chromosomes, including their length, banding pattern, and centromere position. To obtain a view of an individual's karyotype, cytologists photograph the chromosomes and then cut and paste each chromosome into a chart or karyogram. Figure 7.7. This karyogram shows the chromosomes of a female human immune cell during mitosis. Career connection. Geneticists use karyograms to identify chromosomal aberrations. The karyotype is a method by which traits characterized by chromosomal abnormalities can be identified from a single cell. To observe an individual's karyotype, a person's cells, like white blood cells, are first collected from a blood sample or other tissue. In the laboratory, the isolated cells are stimulated to begin actively dividing. A chemical is then applied to the cells to arrest mitosis during metaphase. The cells are then fixed to a slide. The geneticist then stains chromosomes with one of several dyes to better visualize the distinct and reproducible banding patterns of each chromosome pair. Following staining, chromosomes are viewed using bright field microscopy. An experienced cytogeneticist can identify each band. In addition to the banding patterns, chromosomes are further identified on the basis of size and centromere location. To obtain the classic depiction of the karyotype, in which homologous pairs of chromosomes are aligned in numerical order from longest to shortest, the geneticist obtains a digital image, identifies each chromosome, and manually arranges the chromosomes into this pattern. At its most basic, the karyogram may reveal genetic abnormalities in which an individual has too many or too few chromosomes per cell. Examples of this are Down syndrome, which is identified by a third copy of chromosome 21, and Turner syndrome, which is characterized by the presence of only one X chromosome in women instead of two. Geneticists can also identify large deletions or insertions of DNA. For instance, Jacobson syndrome, which involves distinctive facial features as well as heart and bleeding defects, is identified by a deletion on chromosome 11. Finally, the karyotype can pinpoint translocations, which occur when a segment of genetic material breaks from one chromosome and reattaches to another chromosome or at a different part of the same chromosome. Translocations are implicated in certain cancers, including chronic myelogenous leukemia. By observing a karyogram, geneticists can actually visualize the chromosomal composition of an individual to confirm or predict genetic abnormalities in offspring even before birth. Non-disjunctions, duplications, and deletions. Of all the chromosomal disorders, abnormalities in chromosome number are the most easily identifiable from a karyogram. Disorders of chromosome number include the duplication or loss of entire chromosomes, as well as changes in the number of complete sets of chromosomes. They are caused by non-disjunction, which occurs when pairs of homologous chromosomes or sister chromatids fail to separate during meiosis. The risk of non-disjunction increases with the age of the parents. Non-disjunction can occur during either meiosis 1 or 2 with different results. If homologous chromosomes fail to separate during meiosis 1, the result is two gametes that lack that chromosome and two gametes with two copies of the chromosome. If sister chromatids fail to separate during meiosis II, the result is one gamete that lacks that chromosome, two normal gametes with one copy of the chromosome, and one gamete with two copies of the chromosome. Figure 7.8. Following meiosis, each gamete has one copy of each chromosome. Non-disjunction occurs when homologous chromosomes, meiosis I, or sister chromatids, meiosis II, fail to separate during meiosis. An individual with the appropriate number of chromosomes for their species is called euploid. In humans, euploidy corresponds to 22 pairs of autosomes and one pair of sex chromosomes. An individual with an error in chromosome number is described as aneuploid, a term that includes monosomy, loss of one chromosome, or trisomy, gain of an extraneous chromosome. Monosomic human zygotes missing any one copy of an autosome invariably fail to develop to birth because they only have one copy of essential genes. Most autosomal trisomies also fail to develop to birth. However, duplications of some of the smaller chromosomes, 13, 15, 18, 21, or 22, can result in offspring that survive for several weeks to many years. Trisomic individuals suffer from a different type of genetic imbalance, an excess in gene dose. Cell functions are calibrated to the amount of gene product produced by two copies, or doses, of each gene. Adding a third copy, or dose, disrupts this balance. The most common trisomy is that of chromosome 21, which leads to Down syndrome. Individuals with this inherited disorder have characteristic physical features and developmental delays in growth and cognition. The incidence of Down syndrome is correlated with maternal age, such that older women are more likely to give birth to children with Down syndrome. Figure 7.9. The incidence of having a fetus with trisomy 21 increases dramatically with the age of the birth parent. Humans display dramatic deleterious effects with autosomal trisomies and monosomies. Therefore, it may seem counterintuitive that human females and males can function despite carrying different numbers of the X chromosome. In part, this occurs because of a process called X inactivation. Early in development, when female mammalian embryos consist of just a few thousand cells, one X chromosome in each cell inactivates by condensing into a structure called a Barr body. The genes on the inactive X chromosome are not expressed. The particular X chromosome that is inactivated in each cell is random, but once the inactivation occurs, all cells descended from that cell will have the same inactive X chromosome. By this process, females compensate for their double genetic dose of X chromosome. In so-called tortoiseshell cats, X inactivation is observed as a coat color variegation. Females heterozygous for an X-linked coat color gene will express one of two different coat colors over different regions of their body, corresponding to whichever X chromosome is inactivated in the embryonic cell progenitor of that region. When you see a tortoiseshell cat, you will know that it has to be female. Figure 7.10. Embryonic inactivation of one of two different X chromosomes encoding different coat colors gives rise to the tortoiseshell phenotype in cats. In an individual carrying an abnormal number of X chromosomes, cellular mechanisms will inactivate all but one X in each cell. As a result, X chromosomal abnormalities are typically associated with mild intellectual and physical disabilities, as well as sterility. If the X chromosome is absent altogether, the individual will not develop. Several errors in sex chromosome number have been characterized. Individuals with 3 X chromosomes, called triplo X, are assigned female but express developmental delays and reduced fertility. The XXY chromosome complement, corresponding to one type of Klinefelter syndrome, corresponds to male individuals with small testes, enlarged breast, and reduced body hair. The extra X chromosome undergoes inactivation to compensate for the excess genetic dosage. Turner syndrome, characterized as an XO chromosome complement, meaning only a single sex chromosome, corresponds to a female individual with short stature, webbed skin in the neck region, hearing and cardiac impairments, and sterility. An individual with more than the correct number of chromosome sets, 2 for diploid species, is called polyploid. For instance, fertilization of an abnormal diploid egg with the normal haploid sperm would yield a triploid zygote. Polyploid animals are extremely rare, with only a few examples among the flatworms, crustaceans, amphibians, fish, and lizards. Triploid animals are sterile because meiosis cannot proceed normally with an odd number of chromosome sets. In contrast, polyploidy is very common in the plant kingdom, and polyploid plants tend to be larger and more robust than euploids of their species. Chromosome structural rearrangements. Cytologists have characterized numerous structural rearrangements in chromosomes, including partial duplications, deletions, inversions, and translocations. Duplications and deletions often produce offspring that survive but exhibit physical and mental abnormalities. Cri du chat, from the French for cry of the cat, is a syndrome associated with nervous system abnormalities and identifiable physical features that result from a deletion of most of the small arm of chromosome 5. Infants with this genotype emit a characteristic high-pitched cry upon which the disorder's name is based. Figure 7.11. This individual with Cri du chat syndrome is shown at various ages: A, age 2, B, age 4, C, age 9, and D, age 12. Chromosome inversions and translocations can be identified by observing cells during meiosis because homologous chromosomes with a rearrangement in one of the pair must contort to maintain appropriate gene alignment and pair effectively during prophase 1. A chromosome inversion is the detachment, 180-degree rotation, and reinsertion of part of a chromosome. Unless they disrupt a gene sequence, inversions only change the orientation of genes and are likely to have more mild effects than aneuploid errors. Evolution connection. The chromosome 18 inversion. Not all structural rearrangements of chromosomes produce non-viable, impaired, or infertile individuals. In rare instances, such a change can result in the evolution of a new species. In fact, an inversion in chromosome 18 appears to have contributed to the evolution of humans. This inversion is not present in our closest genetic relatives, the chimpanzees. The chromosome 18 inversion is believed to have occurred in early humans following their divergence from a common ancestor with chimpanzees approximately 5 million years ago. Researchers have suggested that a long stretch of DNA was duplicated on chromosome 18 of an ancestor to humans, but that during the duplication it was inserted into the chromosome in reverse orientation. A comparison of human and chimpanzee genes in the region of this inversion indicates that two genes, ROC1 and USP14, are farther apart on human chromosome 18 than they are on the corresponding chimpanzee chromosome. This suggests that one of the inversion breakpoints occurred between these two genes. Interestingly, humans and chimpanzees express USP14 at distinct levels in specific cell types, including cortical cells and fibroblasts. Perhaps the chromosome 18 inversion in an ancestral human repositioned specific genes and reset their expression levels in a useful way. Because both ROC1 and USP14 code for enzymes, a change in their expression could alter cellular function. It is not known how this inversion contributed to hominid evolution, but it appears to be a significant factor in the divergence of humans from other primates. A translocation occurs when a segment of a chromosome dissociates and reattaches to a different non-homologous chromosome. Translocations can be benign or have devastating effects depending on how the positions of genes are altered with respect to regulatory sequences. Notably, specific translocations have been associated with several cancers and with schizophrenia. Reciprocal translocations result from the exchange of chromosome segments between two non-homologous chromosomes, such that there is no gain or loss of genetic information. Figure 7.12. In A, an inversion occurs when a chromosome segment breaks from the chromosome, reverses its orientation, and then reattaches in the original position. B shows a reciprocal translocation occurring between two non-homologous chromosomes and does not cause any genetic information to be lost or duplicated.